Good morning, everyone. Good morning. Go ahead and type your name into chat for me if you would. Thank you. Get a little more light there. Thank you very much. Yeah, just type your name into chat for me as usual. Thank you. Okay. So today's exam day, your lecture exam is today. I saw that a few of you have already taken it. Good for you. Just remember, you need to get that submitted to me by 8 o'clock tonight. And hard to believe that'll be your third lecture exam. All done. As I mentioned to you last time, we're moving into, I think, some of the most difficult material in the course, which is metabolism. Next week and the week after, we're going to be talking about, in lecture, we're going to be talking about very complex topics, enzymes and the concept of energy, what we mean scientifically when we talk about energy in a cell. We're going to talk about some things related to genetic material. We're going to talk about how DNA gets replicated in cells, particularly microbial cells, about how sometimes that process goes awry and mutations are the result. I'm going to talk about what we know as gene expression. And when we use that phrase, gene expression, what we're talking about is a gene, a piece of DNA, being used to build a protein. The gene gets expressed, it gets used. And the result, through transcription and translation, is a protein, a functional protein. Those are very complicated, detail-rich subjects, and they're challenging. So be sure you give yourself the time and the attention that those complex topics deserve. In laboratory, we're moving on today to talk a little bit about how we control microbes. We've talked about how we can keep track of microbes in our world. We talked about how we can sample various environments around us, and we can keep track of what's growing. What kind of microbes are growing in our buildings and in our ventilation systems and in our water bodies and so on and so on. Today, we're going to talk a little bit about one of the ways that we can control the growth of microbes. We know they're there, we know they can be problematic, and at times we need to take steps to clean our environments and control the number and the types of microbes that are around us. We're never going to get rid of microbes totally in our environment. It's just not going to happen. But we can take steps to reduce the prevalence of pathogens, especially. And one of the ways we do that is through physical methods. So in other words, not chemical. Today we're talking about one of the physical methods that we use. Remember, when I say physical methods, I'm talking about things like heat and radiation. Physical methods that we can use in our environment to either stop the growth of cells or to kill them outright. And the one we're going to focus on today is ultraviolet radiation. Ultraviolet radiation is an easy, inexpensive, effective way to control the growth of microbes in certain environments, not in all situations. We're going to talk about why ultraviolet is limited in its application. But it's effective. You and I know that the sun produces ultraviolet radiation, among other types of electromagnetic radiation. The sun is a source of ultraviolet light for us. And we know that ultraviolet light is dangerous to us, right? We know that now. We know that in 2021. We know that we have to cover our skin with sunscreens when we're out in the sun because these are powerful types of radiation, these ultraviolet rays. They can damage our skin and they can bring on skin cancers. They can damage our eyes and bring on cataracts later in life. Ultraviolet radiation is powerful. It has strength behind it. And we're going to harness it. We do harness it, again, in certain situations in order to control the microbes that sometimes cause us problems in the world around us. So before we jump into the laboratory exercise for the day, I'm just going to pull up our schedule and we could take a look at what's coming up next week. Just as a reminder of things, it's always good to get extra reminders. At least I know I like to be reminded again and again. I have to write everything down in my life or I won't remember it. Okay, so that's what you should be seeing on your screen right now, our home page in Canvas. I'm just going to click on modules, and then I'm going to scroll down to our current module, which is right here. So in today's lab, we're going to look at controlling the growth of microbes with ultraviolet radiation. And of course, after today, you'll be able to work on and complete your lab homework for the week, which as usual is due Sunday by midnight. Just a reminder that that's July 4th. So if you have any plans, fireworks or whatever planned for July 4th, you'll just want to get that in early for me. Okay, so looking ahead to next week, here you can see those difficult lecture topics that I mentioned. Microbial metabolism, part 1, that's the lecture about enzymes and how enzymes function and this concept of energy. And then for the second lecture topic, we have cellular respiration, oxidative phosphorylation, and fermentation. And of course, a lecture quiz to go with each of those. Now we'll meet on Monday for lab, and we will continue talking about controlling the growth of microbes, but we're going to move on to chemical methods that we use to control microbes in our environments. So that will be on Monday. Take note that there is no lab on Wednesday next week because you have your next lab practical exam. So that's coming up next week. We're moving forward in the course. We're really right in the meat of the course now. Take a moment when you have one to look on Canvas at your grades. Take a moment to pull your grades up and scroll down Make sure that you haven't accidentally forgotten to submit an assignment or accidentally forgot, to look at some comments that I might have left for you on an assignment. Take some time to do that. This is a good time to take stock of where you are grade-wise in the course. You always have access to that in Canvas. You always have access to how many points you've earned out of the available points that have been offered so far. And of course, if you find that you are not doing as well right now as you would like to do, if you feel like you started off strong and maybe you're slipping a little bit now as the subject matter gets harder and harder, take some time to think about how you might change it up a little bit. Sometimes at the beginning of a course like this, when it's largely review, you don't have to put in as much time. But then as we get into harder and more unfamiliar topics, you may need to spend some more time watching these lecture videos. You may need to watch parts of a lecture video more than once. You may need to go into your textbook and read a little more about one of these subjects that we talk about in lecture because it's not making sense to you. Give yourself that time. You've been working hard so far, so don't let up. Don't let up now. If you've earned a good grade so far, hold on to that good grade and give the coursework the time that it needs. Okay? Just my little advice for mid-course evaluations of your work. All right. Does anybody have any questions about anything we talked about last time? Does anybody have any questions about the oxygen category material that we talked about the last time we were together? Or questions about lecture material or lecture quizzes or anything like that? I noticed that most of you did really well on the lecture quiz that was due last night. Most of you did very well on it, perfect score. So good on you. You're doing good. All right. All right. I'm going to go ahead and open up our slides for today, and we'll start talking about this very powerful tool we have in the management of microbes called ultraviolet radiation. And that's what you should be seeing on your screen right now, the title slide for Controlling Microbial Growth with Ultraviolet Light. On this slide, you're looking at a list of our objectives for our meeting today. We want to talk first about ultraviolet radiation and the features of UV radiation. Ultraviolet is just one of the types of electromagnetic radiation that we experience as inhabitants of planet Earth. We're going to talk about how ultraviolet radiation affects bacterial cells specifically, but we could apply this to other microbial cells as well. We're going to talk about how ultraviolet exposure can both damage cells by damaging their DNA and also kill cells if they get exposed to enough ultraviolet over a long enough period. We're going to talk about how ultraviolet light damages DNA in living cells, because again, even though we're thinking about controlling microbial growth, in reality, all living cells are susceptible to the effects of ultraviolet radiation. We'll talk about how UV exposure damages DNA and how that damage can then lead to mutations in the cell. And finally, we're going to talk about two really important metabolic pathways inside of living cells that repair DNA damage. Through evolution, there are pathways of repair that have evolved to address this problem of DNA damage. DNA gets damaged in living cells. It often gets damaged during the process of copying DNA. It's a natural process that sometimes goes awry. And living cells have developed several means of repairing that damage. You and I have DNA repair systems in our cells, and so do microbes. We're going to specifically talk about two of those repair pathways today. We're going to talk about what's called photoreactivation, or light repair, as it's sometimes called. And we're going to talk about excision repair, or what's also known as dark repair. We'll do an experiment today where we expose bacteria to ultraviolet light, and we'll look to see whether or not one or both of these pathways can be activated in order to repair exposed bacterial cells. To begin today, though, I want us to think a little bit about what ultraviolet radiation is and where it sits in this spectrum of electromagnetic radiation. What you're looking at on this slide is a spectrum of all the different kinds of electromagnetic radiation that we are exposed to on this planet. I want you to notice there's a little sliver of space right here in the spectrum, and the artist has expanded that space for us to show us that this little sliver right here is where visible light fits in the electromagnetic radiation spectrum. So the light that we are able to see, the light that we are able to process in our nervous system through our eyes, this is visible light, and visible light is simply another type of electromagnetic, or EM, radiation. You'll notice that there is a wavelength listed here, running from about 400 for the purple blue end of visible light, up to 700 plus for the red end of visible light. When we talk about EM radiation, we always talk in terms of wavelength, because that's how light is measured in terms of its power, in terms of its strength. So if we go to one end of this spectrum, where you can see gamma rays, you'll notice that gamma rays have the most energy, 10 to the 8th electron volts. And they also have the shortest wavelength, 10 to the minus 14th meters. So remember, when we talk about light, if you remember from physics, electromagnetic radiation and visible light, it has this really unusual property. It travels both as a wave and as a particle, right? Remember this from physics? And we can measure the wavelength of different kinds of electromagnetic radiation, from the peak of one wave to the peak of the next wave, or from the low point of one wave, the trough, to the low point of the next wave. That's the wavelength. What we know is that the shorter that distance is, in other words, the shorter the wavelength, the more powerful that EM radiation is. Now, what do we mean by power? Well, we mean it's capable of penetrating through materials, including living things. So if we go to that one end of the spectrum where gamma rays are, And of course, gamma rays are the kind of EM radiation we associate with things like nuclear power and nuclear bombs. Gamma rays can penetrate almost anything, right? They certainly penetrate our bodies and damage our cells. Gamma rays have a lot of power and a very short wavelength. Now, if you go to the opposite end of the spectrum, you find EM that has a very long wavelength and a very low amount of power. Let's take a look at that. So all the way at the other end of the spectrum, that's where radio waves are. You can see that when it comes to FM radio and AM radio, the AM has the least amount of power associated with it or energy associated with it and the longest wavelength. Then you have shortwave radio. Then you have television signals, FM. We're talking about very long wavelengths and very low amounts of energy. Now, if we keep going along this scale, so the wavelength is getting shorter and shorter. You come into radar, then you come into infrared, then you hit visible light. And again, visible light, we're talking about a wavelength of between 400 and 700 nanometers. Okay. Now we're leaving visible light and we're moving into more powerful, more energetic wavelengths. Ultraviolet is right there. So it's more powerful than visible light, more energetic, shorter wavelength. Then we get into X-rays, then we get into gamma rays. So the shorter the wavelength, the more energetic we're talking about. It's all electromagnetic radiation. It's all the same. It's all electromagnetic radiation, but it takes different forms depending on its wavelength and how energetic it is and how capable it is of harming cells because it penetrates those cells and damages them on the way through. So visible light is not harmful to us. And anything over to the right in that spectrum, not harmful to us because it can't penetrate through our tissue. Radio waves can't penetrate through your tissue. Radar, infrared, visible. But once you get ahead, once you move into a shorter wavelength past visible, now you're talking about EM that can harm you. And ultraviolet is the first one we run into. All right. Now, ultraviolet radiation is what we refer to as non-ionizing radiation, and that's important because the other two types of EM radiation on that spectrum, the X-rays and the gamma rays, those are ionizing. So UV is very powerful, but it is not ionizing. Now, what does that mean? It means that as it passes through living tissue, it's not strong enough to knock electrons out of orbit. That's what ionizing radiation can do. When ionizing radiation encounters atoms inside of living cells, it's powerful enough to knock electrons out of orbit. And as we've talked about in here, when an atom has an unpaired electron in its valence shell, it's in trouble. It's become something dangerous because it's unstable. Remember, those are what we call radicals or free radicals. They are missing one of their electrons and they will go on a hunt to get another electron. Ionizing radiation knocks electrons out of orbit. Ultraviolet, though, cannot do that. It's not powerful enough. Now, when it comes to the actual wavelength of UV, we're talking about a range. So just like visible light, ultraviolet does not have one wavelength. It has a range of wavelengths. It ranges from 15 up to 400 nanometers in wavelength. Think about how small that is. Remember, nanometers. There are 1 million nanometers in a millimeter. So it's a very small wavelength. Remember, visible light starts at 400 and goes to 700. Ultraviolet is right below there, 15 to 400 nanometers in wavelength. Now, within this range of wavelengths, we can break this down into 3 different types of ultraviolet. Now, you've probably heard these terms. We refer to these as UVA, UVB, and UVC. If you've ever picked up and read the label on a bottle of sunscreen, you've heard these terms before. Let's look at what they mean in terms of wavelength and energy. From a wavelength of 200 to 290, right here, we're talking about the most damaging type of ultraviolet. This is ultraviolet C. And this is what's considered biocidal. This will kill living tissue if it's exposed long enough. It creates the most damage. And that's because in this spectrum, 200 to 290 nanometers, this is the optimal wavelength for absorption by DNA. Our DNA molecule will absorb ultraviolet the best when it runs between 200 and 290 nanometers in wavelength. Now you might say, well, what about 15 up to 200? What about the lower end of the spectrum there? Well, it turns out that ultraviolet radiation that has the very lowest wavelength, the very shortest wavelength, the 15 to 200 rays, they don't even come down and interact with us on the Earth. They typically get absorbed by molecules that are in the air. That's good news for you and I, because this would be the most powerful type of ultraviolet. It doesn't even reach the ground. Once you hit 200 nanometers, now you're talking about ultraviolet C, and that is going to come down to the ground and interact with living things. Biocidal, capable of damaging and even killing cells. Now, I do want to point out that there's a note on this slide about a figure. This is in the laboratory manual that we sometimes use for this course. You guys did not purchase that manual, so go ahead and just eliminate that little note here. Now, once you get a little bit bigger, once the wavelength gets longer than 290, now you're talking about UVB. So from 290 to 320 nanometers wavelength, you're talking about UVB. This type of ultraviolet can also damage DNA, but it is less powerful than UVC. It's interesting because this type of ultraviolet, UVB, is the type that actually has a health benefit for you and I. Ultraviolet rays that are about 300 nanometers in wavelength are the type that help convert vitamin D1 into vitamin D3 in our skin. You might have learned about that in some of your earlier courses. Ultraviolet light can help create active vitamin D in our skin. You need about 15 minutes of exposure each day in order to make your own active vitamin D3. Now, finally, we have UVA, which ranges from 320 to 400 nanometers in wavelength. UVA is not readily absorbed by DNA. So this is a type of ultraviolet that is not causing the DNA damage in living cells. I don't have it written on the slide, but I will mention that for humans, you still don't want unlimited exposure to UVA rays, because even though they can't damage the DNA in your cells, in your skin cells, they can damage the molecules in your skin in the sense that they can cause what we would consider premature aging. So a lot of people are concerned about how wrinkled their skin is and how it looks as it ages. Even though UVA is not going to damage the DNA in your skin cells, it will damage other molecules and bring on those changes that we associate with sun damage. So that's why sunscreens, depending on the brand, will protect against UVA as well. So UVA can age your skin, but it's not damaging the DNA in skin cells. The same thing is happening, if you think about it, to microbial cells. When they get exposed to UVA, it's not damaging to them because it's not being absorbed by their DNA. And I'm pretty sure they're not too worried about, about aging, our microbial friends. When you get into UVB, now you have a wavelength of ultraviolet that's capable of damaging DNA. And when you get into UVC, now you're talking about a wavelength of ultraviolet that can damage DNA and, with enough exposure, can kill cells. Now, one of the most important things to know about ultraviolet is this. Because of its relatively long wavelength compared to other penetrating forms of radiation, ultraviolet has a huge limitation. It's only able to penetrate about 2 millimeters deep. Let me say that again, because that's super important. The number one limitation to ultraviolet as a means of controlling the growth of microbes is that it does not penetrate material. So if I'm using it because I want it to kill microbial cells, I have to remember that. It can only penetrate to a depth of about 2 millimeters. That's why exposure to the sun, while it can damage your skin and it can damage your eyes, your cornea, it can't damage your liver, it can't damage your heart because it can't reach. It can't get that deep inside of you. It can penetrate to 2 millimeters approximately. Now, if you're a microbe, 2 millimeters is an enormous depth, right? If you're a microbe, 2 millimeters is plenty. The ultraviolet will go right through you because, of course, microbes are in the micrometer range, not the millimeter range. They're in micrometers of diameter. So, of course, ultraviolet goes all the way through a microbe. But it's good news for you and I that it doesn't penetrate any deeper than it does. Okay. So ultraviolet radiation has some use to us in controlling the growth of microbes in our environment. It is a really nice disinfectant on smooth, hard surfaces. Put a star right next to that. Smooth, hard surfaces. Because it has one huge drawback, and that's poor penetration. Ultraviolet can penetrate to about 2 millimeters of depth. So any cell that it passes through in that 2 millimeters is going to get its DNA damaged by the UVB and UVC wavelengths. And if it gets enough exposure, the UVC is going to kill it. For us, our skin and our cornea are most vulnerable to ultraviolet, which is why we wear sunscreens and we wear the appropriate eyewear, sunglasses, when we're out in the sun. Now, human beings have evolved a little bit of a protective mechanism, haven't they? If you remember from your anatomy classes, human beings have a molecule called melanin in their epidermis, right? And melanin, which is a pigment, can absorb ultraviolet. That does offer us some protection. And that's why when we're out in the sun and getting exposed to UV, our skin cells start to produce more and more melanin as a protective measure. That's why we tan when we're out in the sun. But obviously melanin has its limits, right? Melanin has its limits. And the reason our skin burns over time is a combination of the heat of the sun and the damaging ultraviolet rays. Microbial cells don't have any melanin. Microbial cells are completely vulnerable to ultraviolet. There are two things that a microbial cell experiences from exposure to ultraviolet. The DNA gets damaged, and that's going to impact just the health of that cell if its DNA isn't right. And if enough damage is accrued, the cell's going to die. So when we use ultraviolet to control the growth of cells, this is what we're interested in. We're interested in killing those cells so they no longer exist in the environment. Let's talk about what ultraviolet is doing to the DNA molecule. Remember, the DNA molecule that's in our cells is the exact same molecule that's inside microbial cells. The exact same structure, the same 4 bases. It's the same molecule and it suffers the same fate when it gets exposed to ultraviolet. On this slide, you're looking at a drawing of the double helix of DNA, and the artist has drawn the different bases with different colors. So A, G, C, and T are represented by these colored horizontal bars, with thymine, or T, being the yellow bar. The way ultraviolet light damages DNA is by damaging this thymine base. And very specifically, any place along the length of the DNA molecule where there are 2 thymines side by side, any place along the DNA molecule that there happen to be 2 thymines side by side, what the ultraviolet is going to do is cause those 2 thymines to bind to each other. That creates what we call a thymine dimer. Take a look at this little drawing down here. This pink line right here represents the backbone of one strand of DNA. And you can see there are 2 bases jutting off of that DNA. These are both thymines, hence the TT. When ultraviolet hits those thymines, it's going to cause them to bind to each other. So they're no longer hydrogen bonding with their base pair. And of course, for thymine, their base pair would be adenine on the other strand. Instead of binding to 2 adenines, they're going to essentially turn and bind to each other. So that creates this mess, as you can see in the drawing. Now the 2 yellow bars, they're not reaching across and binding with the green adenines. They're binding with each other. And look what has happened in that area. The double helix is not right anymore. It's broken in that area. The DNA molecule has been damaged. Now, damaged DNA matters. If those two thymines were sitting in the middle of a gene, and that gene needed to be transcribed and translated because a protein was needed, we'd have a big problem, wouldn't we? Because that gene is damaged. It's not going to be available to make protein from. So that's bad enough. But there's another consequence of DNA damage, and that comes when it's time for that cell to replicate, when it's time for that cell to go through binary fission and divide. Remember what happens to the DNA when a cell, a microbial cell, a bacterial cell, for example, needs to divide. The entire DNA molecule needs to be replicated. It needs to be copied. And an enzyme does that work. The DNA polymerase enzyme, as it's called, travels down each strand of DNA and makes a copy. Not an exact copy, right? A complementary copy. Where there's an A, it puts a T. Where there's a G, it puts a C, and so on. When that enzyme gets to that dimer, it doesn't know what to do. When it's time to replicate the DNA and the replication enzyme gets to that dimer, it doesn't know what to do. It doesn't know what to put there because it doesn't look like thymine anymore. It just looks like this blob. So what the enzyme does is it guesses and it just puts any two bases there. Now, that is a mutation. That's a mutation, a change in the order of the bases in the DNA molecule. Where that thymine dimer is, the new strand of DNA that's made from the old strand, the new strand should have two adenines, right? One for each of those thymines. But the enzyme is just going to place some random base there. Maybe two guanines, maybe a guanine and a cytosine, maybe two thymines, who knows? That changes the normal order of the bases. There should be two adenines there. There's probably not going to be two adenines there. Now you have a mutation. So the ultraviolet light itself is not mutating the DNA. That's a small point, but it's an important point. The ultraviolet light is damaging the DNA. It's creating the dimers, the thymine dimers. A natural process that comes later on, DNA replication, creates the mutations. So that's how you and I end up with skin cancer sometimes when we get exposed to ultraviolet light. We get dimers in our DNA, in our skin, in our cornea. We get dimers. And when our cells, when our skin cells are replicated, our replication enzyme doesn't know what to do. And you end up with new DNA that has a different sequence of bases. That's a mutation. And some of those mutations lead to cancers. In a microbe, those mutations are going to change the cell fundamentally. Some mutations don't have any effect at all. Some mutations are actually helpful to a cell because they give the cell some new trait that turns out to be helpful. That's the basis of evolution. But some mutations are very damaging. Some mutations are so damaging, in fact, that the cell will die. So for a microbial cell, exposure to ultraviolet in UVB and UVC ranges is going to cause DNA damage through the formation of those dimers. And it may lead to mutations down the road. If they're exposed to enough, in other words, for a long enough period of time, enough UV, there'll be so much damage in that DNA molecule that the cell will die. So bad news for the microbe, but good news for us if we're trying to kill that cell. Good news for us, we just need to hit it with a bunch of ultraviolet. And if we do it correctly, for a long enough period of time correctly, we'll kill that cell and it won't be a threat to us in our environment. So ultraviolet radiation can damage and or kill microbial cells. But we can't forget that thing I mentioned at the beginning of class today, and that is that living cells have evolved DNA repair mechanisms. Most of the damage that happens in your skin when you get exposed to the sun, most of that damage gets repaired. Those thymine dimers get fixed by other enzymes inside of your cell whose job it is to protect your DNA. Obviously, not all of them get fixed because some people do get skin cancers. But a lot of DNA damage gets repaired. There are several mechanisms of DNA repair. We're not going to talk about all of them. We're going to talk about two of them that are in microbial cells, though. This photoreactivation system, or light repair, and the excision system, or dark repair. So these are the details that I want us to be familiar with regarding these repair systems. Photoreactivation, this photo phrase here, this means light. Anytime you see photo, think light. It turns out that light within the visible range, within visible wavelengths, so between 400 and 700, that exposure of a microbial cell just to ordinary visible light is going to activate a repair enzyme, or I should say a family of repair enzymes. These are called photolyases. Photolyases. And what these enzymes do is they travel along the length of the DNA molecule, and if they come upon a thymine-thymine dimer, they split it. They break the bond that holds the two thymines together. That's going to allow those two thymines to sort of bounce back into their original position, and the normal anatomy of the DNA strand will be restored. So the cell has to be in light in order for these enzymes to be active. Excision repair is a little bit different. Excision repair doesn't require light exposure. The cell does not need to be in the light for excision repair to occur. That's why this is sometimes called dark repair. There's another family of enzymes that does excision repair. These are just known as endonuclease enzymes. This is an enormous family of enzymes. And what an endonuclease does is travel down the strands of DNA, and when it encounters a thymine-thymine dimer, it cuts it out. It removes it completely from the strand. That's going to leave a hole, right? That's going to leave a hole in one strand of the DNA molecule. Other enzymes will come in and repair the hole. They'll put two new thymines down and repair that hole. So two different systems, two different families of enzymes, but the end result is the same. The normal anatomy of that DNA strand is going to be restored. We're going to fix the problem. Sounds great, doesn't it? And these systems, we have similar systems inside of our cells. They work hard. They're very good at repairing our DNA. But here's the important thing to note. None of these repair systems are 100% accurate. In other words, some of these thymine dimers are going to persist. They'll get missed by the enzyme systems, and they'll persist. So we're going to do an experiment. We're going to do an experiment today with a couple of different bacterial species. We're going to look to see what effects we can have on bacterial cells by exposing them to ultraviolet light. But we're not going to stop there. We're also going to incubate those cells after they've been exposed to ultraviolet in order to see if the repair mechanisms kick in. And we'll ask which repair mechanism kicks in too, by incubating some of those exposed cells in the light and some of those exposed cells in the dark. So we'll expose bacterial cells to ultraviolet light for different time periods, because we know the time of exposure is very important with UV. And we'll look first to see whether or not the cells survive that. We'll give them 30 seconds of exposure, and we'll give them 60 seconds of exposure. I know that seems like a very short amount of time, doesn't it? But remember, we're talking about microbial cells. They have no protection from ultraviolet. Now, exposing them isn't enough. We have to expose them to the ultraviolet, and then we have to incubate them to see if they're still alive, right? If they're alive, they are going to grow into a colony. If they're not alive, they're not going to be able to grow into a colony. So we're going to incubate the exposed plates for 24 hours to see whether or not a colony grows. Some of those plates we're going to put into the light because we're going to see if we can turn on the light repair system. And some of the plates we're going to put in the dark to see if we can turn on the dark repair system. Remember, in the light, it would be this photoreactivation system that got turned on, and in the dark, it would be excision repair. Now, because we're not interested in counting colonies or anything, we don't need to do a spread plate here, at least not in the traditional sense. In fact, we're going to put as many cells on these plates as we can get on these plates, because ultraviolet, we already know, is a really good disinfectant. So we're going to make plates with lawns on them. Remember what that means. If you put enough cells onto an agar plate and spread them all over the surface of the agar plate, you're going to get a lawn of growth. The whole surface of the plate will be covered in growth. And you get this lawn of bacteria. So, in other words, we're not looking originally, if we took this plate, in other words, and we didn't expose it to UV, our control, we're going to expect not to see colonies growing, but just a lawn of cells growing. Now, the reason we do that, the reason we put so many cells on the plates is because of how effective UV is. And because that will allow us to tell which cells survive. If I cover the whole plate in cells, and I kill most of those cells, but a few of them survive because of the repair systems, I'll get colonies growing where the cells survived. So we're going to use regular old nutrient agar plates, and we're going to spread bacterial cells out on them. We need to have a control, though, don't we? We need to know that we have a real effect from the UV. We need to be able to show that when we make a lawn and we don't expose it to UV, we're going to get that beautiful lawn growing. When we spread tons of cells out on the plate and we don't expose it to UV, and we put that plate in the incubator, 24 hours later, we're going to have a lawn of cells growing. We need to be able to compare that to the plate that does get exposed to UV. And there's an interesting way that we can do that. We can have a control and a treatment on the same plate. And I'll show you how we're going to do that. These are the organisms that we're going to be using today. The first one is called Bacillus licheniformis. This bacillus is a gram-positive rod. It is a non-pathogen. It happens to be a saprophyte, just like some of the fungi that we talked about. This is a bacterium that's a saprophyte. It lives in the soil and it degrades, it consumes and degrades dead things out in the soil. So there are bacterial saprophytes, just like there are fungal saprophytes. Because it's a bacillus, it's also a spore former. And because it's a spore former, it's a pretty hardy organism, right? When the environment gets inhospitable, it tends to sporulate, and that allows it to survive some really bad conditions that would kill other bacteria. Now, take a look at Bacillus licheniformis when you make a quadrant streak plate. Take a look at these colonies. You can see that the original quadrant, the first quadrant is right here. This is where the cells were originally streaked out. Then some cells were pulled into the second quadrant here. Some cells were pulled into the third quadrant here. You got some colonies in the third quadrant. And then the fourth quadrant over here. Now I want you to notice this colony morphology because it's pretty unusual, isn't it, compared to what we've seen so far. Let me outline for you that this is a colony right here. This is a colony right here. This is a colony right here. A couple things to note. First of all, each colony looks unique. They don't have a uniform size, do they? They also don't have a nice smooth margin around the edges. They're also really big, aren't they? Remember, not all colonies look like nice little round circular structures. Some colonies are enormous. Some colonies are very irregular looking. Some people refer to that as sort of a fried egg appearance because it looks a little bit like the way an egg looks when you fry it up in a pan. So it's important to remember that colony morphology really varies from one organism to another. So this is our bacillus. Let me put this picture away. And we'll take a look at our second organism, which is our old friend E. coli. Remember, E. coli is a gram-negative rod. E. coli lives inside all of us all the time and doesn't cause us any problems, but some strains are pathogenic. E. coli is an enteric coliform. It's an intestinal organism that is capable of fermenting lactose. That's why it's called a coliform. And this is a quadrant streak plate with E. coli on it. This looks much more like what we've seen so far. Here's quadrant 1 and quadrant 2 and quadrant 3 and quadrant 4. And we were successful here. We got individual colonies growing. These are much more traditional, what we would expect to see from a lot more organisms. Relatively small colonies, kind of a beigeish white color. Nice even margin. A lot of bacteria have this type of colony morphology. So, two organisms that come from two very different worlds, right? One of them lives inside of other organisms. That's the E. coli. One of them lives out in the world. One of them lives out in the soil. Two different organisms. Now, here's how we're going to do this experiment. We're going to use nutrient agar plates. we're going to divide these nutrient agar plates into halves. We're going to draw a line down the back of the plate to create two compartments on the plate. One half of the plate is going to get exposed to ultraviolet, and the other half of the plate will not be exposed to ultraviolet. And you might say, well, how are you going to do that? It's actually really easy. All you need is a piece of paper, a nice thick, well, relatively thick piece of paper, because ultraviolet cannot penetrate. It's only capable of penetrating about 2 millimeters. So we just take a piece of paper, just you could use an index card, or you could use Bilbus paper, anything you had available. If you just lay it over half of the plate, those cells are protected. They're protected from the UV exposure. So it's kind of easy. We get to create a control and a treatment on one plate. We can make a direct comparison between those two sides because they all were the same group of cells, and they all grew together in the incubator. They were all in the same conditions except for one difference, whether they were covered or whether they were not covered. So each plate will do this. Each plate we make will cover half and will expose half. Now remember, the other thing we want to do is examine these repair systems. So we're going to make multiple plates for each organism. We're going to spread out pure broth cultures of both Bacillus licheniformis and E. coli, but we're going to use a particularly dense culture. So even though these are technically spread plates in the sense that we're using hockey sticks to spread the cells evenly over the surface of the plate, we're using such a high density of cells that we should get a lawn of growth in our controls. So 2 organisms, 4 plates for each organism. And that's because we have 2 exposure times and 2 incubation conditions. The first plate will get 30 seconds of exposure, and it will be incubated in the light. The second plate will get 30 seconds of exposure, incubated in the dark. See? The next plate will get 60 seconds of exposure, incubated in the light, 60 seconds of exposure, incubated in the dark. So because these plates aren't right in front of you, it's important to visualize something. Remember, when we take a loop of culture, or we take a small volume out of a culture container and we put it onto a plate, and we spread the cells around with a hockey stick. When you look at that plate, you can't see anything, right? The cells are there, you've spread them out, but you haven't given them any time to grow yet. So we spread these cells out. We don't see anything, not yet. We expose that plate to UV right at that point. Then after the exposure, we put the lid on, we put the plate in the incubator, we let it grow for 24 hours. Okay, so it's important to visualize that in your head, because sometimes I think where students get confused in this experiment is they're imagining a plate that already has growth on it getting exposed to ultraviolet. No. We've created a plate with cells on it, yeah. But you can't see those cells, they're microscopic. We spread the cells out, we expose it to UV, then we put it in the incubator, in the light or in the dark, and we see what can grow. If the cell is alive afterwards, it will grow. If the cell is not alive, it won't grow. And the next day, there'll be an empty place on the plate because nothing grew. Okay? All right. So 4 plates, for each organism. Up here on the upper left, you're looking at just a drawing of this spreading process. We've seen this before. We grow up some bacterial culture. We take a small volume of that culture and put it onto the plate. Then we use a sterile hockey stick to spread those cells evenly over the plate. We know that this is a culture that has lots and lots and lots of cells in it because we can see that by how cloudy or opaque the culture is. So we would expect that if we took this plate and we put it in the incubator, we'd get a lawn of growth. Now, in this case, we're not going to expose the whole plate to UV. We're going to do this. Here's the plate. It's been spread with cells. And all we need to do is lay an index card over half of the plate. These cells on this side are going to get exposed. These cells on this side are not. Now notice there's no cover on this. You have to work quickly in this experiment because we have to take the cover off of the plate. Why? Why can't I just leave that plate covered while I do this? Why can't I leave the cover on the Petri dish and then lay my index card over half and then expose it to UV light? Why do you think? I mean, it would certainly be a better move aseptically, right? To leave a cover on top of that agar plate. But I'm taking the cover off. Why? Why would I do that? Connie's got it. Carrie's got it. Because UV can't penetrate. UV can't penetrate through that. It can't go through that cover and hit the cells. So if I left the cover on, that plate, none of the cells would get exposed to UV because the cover would stop that exposure. Ultraviolet doesn't penetrate. I have to have the cells in direct contact with the ultraviolet. Okay, so I have to take the cover off, even though it goes against everything I know about aseptic technique. I have to take the cover off. Now, the other question you might have is, how do you do the UV? And the answer is, we have a little device for that in the laboratory because we use UV in the laboratory with bacteria. We have this thing called a light box or an ultraviolet light box, and that's what it looks like right here. It's literally a box. It's made out of metal. You'll notice that it's painted black. There are tight seals around it, so no light can get in. We want to make sure that the only thing these cells are getting exposed to is ultraviolet. This little thing right here that's plugged in, this is the source of the ultraviolet. You'll notice there's also a viewing port here. It has a filter over it, so none of the room light can get in, but it allows you to peek into the box if you need to see what's happening. So this whole cover comes off of the box. We place the plates into the box. We cover what needs to be covered with index cards. Then we put this black lid back on and we turn the UV light on for 30 seconds or 60 seconds. Then the plates come out and we're going to incubate them. Some of them will be incubated in the light and some of them will be incubated in the dark. All right. Does anybody have any questions about the procedure that we're using or why we're doing anything? It's a little confusing because we're asking a couple of different questions, right? We're looking at 30 seconds of exposure and 60 seconds of exposure because we want to know if there's a difference in the exposure length. There ought to be, right? And we're also asking whether or not repair mechanisms get turned on, either light repair or dark repair. So we have two different sets of questions that we're asking. in this experiment. All right, if anybody has any questions while we're going through the results, just type them into chat, okay? All right, so this is what we got from our 30-second plates. 30 seconds of exposure to UV incubated in the light, 30 seconds of exposure to UV incubated in the dark. On the control side of the plate, that was the side that got covered with the index card, we got a lawn of growth. We saw it on both of the 30-second plates. Remember, we have 2 plates that got exposed to 30 seconds, 2 plates that got exposed to 60 seconds for each organism. So our controls worked great. We saw a lawn of growth on the covered side. On the exposed side, we did not get a lawn of growth, but we did get some individual colonies growing. And we saw that on both incubation plates. So we saw colonies of growth when it was incubated in the light, and we saw colonies of growth when it was incubated in the dark. In other words, some of the cells got repaired, right? Some of these cells survived this 30 seconds. Not all of them, but some of them. This is one of the light incubation plates. This is the plate for Bacillus licheniformis. And wow, look at that plate. It's pretty remarkable, isn't it? This is the side of the plate that got covered with an index card. Every cell that we put onto this plate, I mean, I can't say that for sure, but virtually every cell that we smeared onto this plate survived and grew. We don't get colonies because we put too many cells on the plate. And we got a lawn of growth. This is a solid mass of bacterial growth, this thick layer here. But look at the side that got exposed to ultraviolet. I didn't get a lawn here. Remember, every inch of this was covered. So there was a cell occupying every inch of space on this plate. Some of those cells didn't survive. And the way I know that they didn't survive, or at least I should say they were not capable of dividing, is because there's nothing there the next day. There's no growth. But some of the cells did grow, and that's why I got colonies. Some of the cells divided. They survived, they grew, they divided, they made colonies. Some of them didn't. So I see an effect here. I see an effect after 30 seconds of exposure because some cells were not able to divide. The plate is bare. But I also see some repair going on. I can see some repair happening here. How do I know repair happened? Because some of those cells acted like they didn't even get exposed. Some of those cells did what they always do, which is grow and divide and create a colony. Not all of them did, though. There was a bad effect for some of them, but not for all of them. Just shows you that when we incubate in the light, those light repair systems kick in. And the light repair system was able to repair a lot of those cells. All the cells on one side of the plate got exposed to UV and had their DNA damaged. But those repair systems kicked in and fixed the damage for some of the cells, but not for all of them. These are biological systems, these repair systems, and they are not perfect. But they were able to repair some of those cells enough that they were able to grow. So on that plate, we see two things. We see that 30 seconds of exposure was enough to prevent some of those cells from growing. We also see that light repair mechanisms were successful in repairing some of those cells and allowing them to grow. All right, here's the E. coli plate that was put through similar conditions. So 30 seconds of exposure, incubated in the light. That's what you can see right here. This plate looks pretty different, doesn't it? Look at it next to the licheniformis plate. I do have a lawn. I have a lawn of cells growing on the covered side. That's my control. I got some growth on the exposed side, but gosh, I didn't get nearly as much, did I? I didn't get nearly as much as I did for the bacillus. Some of the E. coli cells were repaired, but not nearly as many as the bacillus cells. Now let's think about that for a minute. If the light repair system had not been activated, I shouldn't have seen anything growing on the exposed side. If the light repair systems had not been activated at all, I wouldn't see anything growing on the exposed side. But I got a few colonies to grow. That tells you that the light repair system worked, but it didn't work very well. I mean, only a handful of cells were repaired. Now, why would that be? How could that be? How could it be that Bacillus licheniformis has a light repair system that seems to work better than the E. coli light repair system? They're the same systems, they use the same enzymes. Why did I see a difference in the amount of repair between two species? They're both bacteria. They're just two species of bacteria, two different genuses, two different species. In one, the light repair system worked really well. In the other, not so great. Any ideas? Any ideas why two species would respond so differently? They're both bacteria. Type it into chat if you have any ideas about that. Two bacteria, two different genuses, two different species, but they're both bacteria, and we know they both have these repair systems inside of them. We showed that the repair systems turn on when you incubate them in the light. But for one of them, the repair system seemed to work much better than in the other one. That doesn't make sense. It's the same repair system, isn't it? So Carrie's jumping in. Who else can jump in? Give me some ideas. Are all bacteria the same? How are bacteria different? What do you think? So both Carrie and Lacey have asked about the cell wall, gram-positive, gram-negative, because we know Bacillus licheniformis is gram-positive and E. coli is gram-negative. Think about that for a minute. Should the cell wall of a bacterial cell make a difference in terms of ultraviolet exposure? Can ultraviolet penetrate a bacterial cell wall? Ultraviolet can penetrate 2 millimeters. Is a thick layer of peptidoglycan enough to protect a gram-positive organism? This is all about sizes, right? Those two bacterial cells are 1 to 2 micrometers across, and one of them has a thick layer of peptidoglycan and one of them has a thin layer. Do you think that thick or thin layer could stop ultraviolet? What do you think? Lacey's saying, yeah, maybe, right? Lacey, that's not enough peptidoglycan. Even if peptidoglycan had some kind of an unusual property, let's imagine that peptidoglycan is like a sunscreen. We don't know that, right? But imagine it is. You would have to have a lot of peptidoglycan around that cell in order to stop that ray, because that ray can come through 2 millimeters. So peptidoglycan would either have to be very thick or reflective. And I can tell you it's neither one of those because it's carbohydrate. So it's not the gram status here, although that is a great guess. It's not the gram status here. It's something we haven't really talked about. I mean, this is about evolution, really, right? Think about where these two organisms live. Who gets exposed to more UV? Bacillus licheniformis or E. coli? Generally speaking, as a species, who's getting more UV exposure? Bacillus, right? Bacillus. Listen, E. coli lives in the intestines. E. coli does get out of your intestine from time to time, right? It's a fecal organism. We find E. coli in bodies of water, yeah. But the place that E. coli lives, generally speaking, is in the intestine. Bacillus lives out in the soil. So through millions of years of evolution, remember, bacteria are the oldest organism on this planet. These two organisms have evolved very differently. Yes, they both have a light repair system, but the light repair system works better in Bacillus. And that's because it has to, or Bacillus would not have survived. Bacillus would not be adapted to growth in soil unless it was better able to cope with ultraviolet exposure. It makes sense if you think about it. E. coli still has to be able to deal with ultraviolet exposure just because it lives on the planet. And it's not 100% in the intestinal tract. Sometimes it comes out of the intestinal tract. But E. coli didn't have the same pressure put on it through evolution that Bacillus did. Bacillus had a lot of selective pressure put on it because it spends all of its time out in the world, exposed to the sun. So it makes sense. It makes sense that this Bacillus organism has a light repair system that works a little better than the light repair system in E. coli. They both have it. It works in both of them. We're just seeing a little more of an effect, perhaps, in the Bacillus. It's all just speculation. We're just guessing here. We didn't test this, did we? All right, now here's another question. I'm making you really think about experimental things today. What if I told you we know for sure that there's no evolutionary differences between those two organisms. In other words, is there any other explanation that you can think of that would explain why there are more colonies on that bacillus plate than there are on the E. coli plate after exposure to UV? Is there any other thing that you can think of regarding our experimental design that would explain that? So we're going to ignore our theory about evolution. What else would explain it? Let's say that we know that they have exact similar light repair systems. How come I got more colonies on the bacillus side than the E. coli side? Think about what we did. I took some cells out of the bacillus tube and I spread them on a plate. I took some cells out of the E. coli tube and I spread them on a plate. What might explain the difference in colony number on those two plates? They both got exposed to UV. They both had light repair happening. I'm thinking about something related to something very basic related to our experimental design. So nothing related to metabolism, nothing related to evolution, something about the design of our experiment that might explain why I got more colonies on one exposed side versus the other exposed side. Connie's got it. Connie's got it. I didn't do anything to count how many cells were in each one of those culture tubes, did I? I took a broth culture of bacillus and a broth culture of E. coli. I told you I knew there were a lot of cells in there because both cultures were very opaque. I didn't measure how many cells were in each culture tube, did I? What if I just put many, many more cells on the bacillus plate than I did on the E. coli plate? I would see the same thing, wouldn't I? If it was purely a number of cells issue, it would make sense. If I had 1,000 cells on one plate and 100 cells on the other plate, let's say the repair system was able to repair 10% of the cells, and I'm making that up. On the 1,000 cell plate, that would mean 100 cells would be repaired and I'd get 100 colonies. On the 100 cell plate, that would mean 10 cells would get repaired and I would get 10 colonies. It could be as simple as that. The only way for us to know for sure would be to do some math with our cultures before we started and start with 2 broth cultures that had the same number of cells in them. So I was putting the same number of cells on each plate. I didn't do that. I just grew up some cells. You have to think about these things sometimes in microbiology because you can't see anything. You can't see how many cells you're spreading out. Good job. Good job thinking that through. All right, let's jump back in. Now, some of you who have eagle eyes will notice that I used the exact same images for both the light and the dark plates here. See that? And that's because I didn't have very good luck taking pictures with this experiment, but it doesn't matter, and here's why. So in other words, I used the same plate in this light image and this dark image. And it turns out it doesn't really matter because it turned out at the end of the experiment that there was no difference between the light and the dark plates. In other words, one of the questions we were asking was about the difference in repair. between light repair and dark repair. It turned out there was no difference between the light and the dark repair. So from here on, we don't even need to think about it. We've already answered that question. Both the light repair and the dark repair showed similar results. It was so similar that we don't even need to think about it from this point on. Both repair systems kicked in for both organisms at both time periods, 30 and 60 seconds. Both repair systems worked and saved some of those cells. Okay, so that's why that and the difficulty I had with my photography. So we've looked at the 30-second results. Now let's look at the 60-second results. On the 60-second plates, 60 seconds of exposure to UV that were incubated in the light, 60 seconds of exposure that were incubated in the dark. On the control side, we got a lot of growth. Very good. That's what we would expect. On the exposed side, no colonies. No colonies. Take a look. Bacillus licheniformis, E. coli. We got nothing. We got nothing. 60 seconds of exposure. We doubled the exposure time. We know that light repair and dark repair were working because of the 30-second plates. We saw on the 30-second plates that both repair systems were working. But when we doubled the exposure time to 60 seconds, we overwhelmed it. Remember, ultraviolet light damages DNA. If cells get exposed, if microbial cells get exposed to enough ultraviolet light, in other words, a long enough period of time, the DNA will become so damaged, the cell will not survive. That's because cells need their DNA. Remember, damaged DNA means damaged genes. And if a gene is damaged, the cell cannot make a functional protein from it. And cells are making proteins 24 hours a day, 7 days a week, 365 days a year, around and around and around. So even within 24 hours of time in the incubator, the cells on those 60-second plates, they didn't survive. The repair systems cannot help if the cell doesn't survive. 60 seconds of exposure was enough to kill every cell on both of those plates. It's amazing, isn't it? In 30 seconds, enough cells survived that to be repaired and to come back. 60 seconds, it's already too much. I said it at the beginning of the day today, so I'll say it again. Ultraviolet is a great disinfectant for hard, smooth surfaces. It works great. 60 seconds, that's all you need. 60 seconds, that's amazing, right? Yeah, Connie's saying, it makes me want to put sunscreen on. I know, I feel the same way. Every time I do this lab, I'm reminded of how powerful ultraviolet is and how amazing it is that we have repair systems, because most of us go through life and don't get skin cancer, right? Most of us. It shows you how powerful and how lucky we are that we have repair systems in place. All right. So let's talk through this a little bit more so you'll be all set to do your homework. What effects did 30 seconds of ultraviolet exposure have? We'll start with Bacillus licheniformis. Did it kill all of the cells on the plate? No. No, it didn't. How do I know? Because I saw colonies growing the next day. Some colonies grew on that side of the plate that got exposed to 60 seconds of or 30 seconds of light. In fact, I saw that on both Bacillus licheniformis and E. coli, both plates. I saw colonies. Some of the cells had to have survived. How do I know? Because they got repaired and they grew. Some of them had to have survived because they got repaired and they grew into colonies by the next day. I know that it happened in both the light repair system and the dark repair system because I saw it on both plates. I saw it on both the light and the dark plates. Would I recommend disinfecting a stainless steel workplace in a restaurant with 30 seconds of UV exposure? Would I tell that restaurant owner, oh yeah, you just have to turn on a UV light over your workspace. Give it about 30 seconds. Would I tell them that? Imagine the workspace where they cut up chicken all day and they make food. Am I going to advise them to turn that ultraviolet light up on the ceiling, to turn that on for about 30 seconds each day? No, no, right? Because I know that's not going to be enough. 60 seconds or more, right? I know 60 seconds was effective against this bacillus and this E. coli. But who knows? Maybe there are other bacterial species that are even more resistant to UV. So I would tell them to do even more. I would tell them that they should do several minutes each day, maybe even longer, just to be safe. But yeah, 30 seconds clearly is not enough. Plenty of cells survived 30 seconds of exposure. Not as many on the E. coli plates, but plenty of cells survived. 30 seconds did not kill all those cells. But boy, 60 seconds did. 60 seconds hit the Bacillus licheniformis and the E. coli. 60 seconds left me with no cells alive, nothing to repair. The light and the dark systems had nothing to work with because the cells were dead. Now, I mentioned restaurants, so let's talk a little bit about restaurants. If you go into a restaurant, and none of us have been in too many restaurants recently because of the pandemic, but if you go into some busy restaurants, if you can see the kitchen, and of course in some restaurants you can't, but if you've ever been in like a pizza place or a sandwich shop, you know, and you could peek into the kitchen, there's often an ultraviolet unit somewhere in there. Sometimes the ultraviolet units are up on the ceiling. Sometimes they're in doorways. It depends. It depends on the place. Ultraviolet is a great disinfectant, but it's only for smooth, hard surfaces. And that's because it doesn't penetrate. So if there's bacteria on that stainless steel counter, those bacterial cells are right on the surface because they can't go down into the stainless steel. So I know ultraviolet is going to be effective there. Smooth, hard surfaces. Why smooth? Because when you start getting into textured surfaces, even if they're hard, when you get into a surface that has a lot of texture on it, those cells can get into little nooks and crannies and they can be protected from exposure. And of course, when you talk about soft surfaces, now you've got a penetration problem. My favorite example, and I talk about this in the lecture, my favorite example of this is the advertisement that I saw once for an ultraviolet wand. And you can buy a little ultraviolet wand in catalogs, and they're marketed to people who travel, with the idea being, ew, you don't know what's growing in your hotel room. So buy this wand and take it with you, and you can disinfect your hotel room. But they show these wands being used on things like the beds and the carpets and the pillows. And no, it's not going to work. Bacteria can penetrate pores. Bacteria can get into the layers of the bedding, the layers of the mattress. Bacteria can go through the depths of the carpet. If you wave an ultraviolet light over those soft surfaces, you may kill a few cells, but you're certainly not going to disinfect. And that's because ultraviolet light does not penetrate. It's 2 millimeters at best. And your mattress is what, 12 inches, 14 inches? You're not going to disinfect your mattress with ultraviolet. But it sounds good, doesn't it? It sounds good, which is why these companies advertise and market to people who might be concerned about the cleanliness of their spaces, their hotel rooms. You have to remember that this is a tool that is super effective, but only on smooth, hard surfaces. So you're going to see ultraviolet in hospital spaces. You're going to see it. And that's because it's one of the tools that's used to reduce the number of microbes in the environment in the hospital. In the lecture, I also talk about this device that is available to hospitals now, which is essentially a massive ultraviolet unit that they can wheel in to different spaces in the hospital and turn on. It shines ultraviolet light in 360 degrees. And if you leave that light on for a couple hours inside a hospital room, for example, a patient room, you're going to kill some cells that you might have missed when you went in and cleaned that room with chemicals. There are going to be little places in the room that maybe you missed when you cleaned with disinfecting chemicals. And that ultraviolet light is going to do some good work for you. But no, it can't penetrate through soft materials. It just can't. It doesn't have the energy to penetrate through soft materials. Lacey's saying those wands are on clearance at Walmart. I'm telling you, those wands are so popular. If you look in any of the sort of I'll call them high-end catalogs, like Hammacher Schlemmer is one, and Brookstone is one. These catalogs that sell pretty pricey devices to people. And I get a couple catalogs that are marketed to travelers too, and they're in every single catalog, these wands. First of all, that's a dangerous device because unless the person knows better, those people could be staring at that ultraviolet light and it could be affecting their eyes and their skin. That's a dangerous thing to sell like that. And it doesn't do what they're told it does. I think it's terrible. I think it's terrible that we sell that. Oh, Connie, children, if they got their hands on that wand, children would be shining it at each other. Of course they would. And that's terrible. I hope that thing has stickers on it or something that warns people of the danger. Oh, and they charge people a lot of money for them too. Yes. So, yeah. So the next time you're in your neighborhood pizza place or something, take a little peek around and see if they have any ultraviolet units running. And typically, what a restaurant or a place like that is going to do is run those units, when they're closed. You wouldn't want to have that thing on all the time necessarily, would you? You wouldn't want to have, like, for example, you would never roll one of those ultraviolet units around the hospital while patients were around because it's ultraviolet light. It's dangerous to them. You would want to wait, and that's what they do. Basically, they'll clean out a patient room with chemical disinfectants the way you always would, and then sometimes they'll roll these units in, these ultraviolet monstrous units in, turn them on, and just leave them. They leave them for hours. And because the rays are being directed in every direction in 360 degrees, they're able to get into some of the cracks and crevices that chemical disinfectants might miss. Connie's asking about the cost of running those bulbs. Ultraviolet bulbs are expensive, but I can tell you they've become a lot less expensive over the past maybe 15 or 20 years. If you tried to buy like a commercial grade ultraviolet light, let's say back in the 90s or early 2000s, they were very expensive, but the cost has really come down. Um, the cost of a lot of light fixtures now has come down with the advent of more LED type products, and the ultraviolet bulbs have become much more affordable now, which is good news. Good news for all of us. Yeah. So you're all set now to do your homework. Wear your sunscreen. Yes, wear your sunscreen. And I'm going to let you go. Remember, you have to get your exam into me by 8 o'clock tonight. Don't forget that. Don't let that slip. Remember to get your second lecture quiz submitted to me on time this week. Get your homework in. And I will see you again on Monday morning when we will talk about these chemical disinfectants. All right, everybody, have a good day. Stay cool. It's going to be hot again. And I'll see you next week.